Hey, and welcome back. Come on in and take a seat. Today we're going to talk about something pretty cool. How natural selection leads to antibiotic resistance. Let's begin with a refresher about where diversity comes from. Recall from our last conversation on the cell, genetic diversity exists primarily because errors occur when the cell copies their DNA and reassorts the genetic material, and this produces novel arrangements. The genetic diversity can result in morphological and physiological differences between organisms. Let's look at an example of this type of diversity using puppies.
Why puppies? Well, first, because they're cute and they make me happy, but also because puppies exhibit variation in coat color based on genetic diversity. Parents of puppies contribute genetic information, and while there are common characteristics present in all of their puppies, each puppy can have unique attributes as a result of receiving diverse genetic information. Now that we have reviewed where genetic diversity comes from, let's bring this with us to the conversation about natural selection. Natural selection exists when the environment that an organism lives in favors one particular genetic combination over another. Using our puppy example, let's imagine that these are actually wolf puppies that live in woods with lots of thorny vines. Straight, short hair can easily pass through the thorns, but long, shaggy hair would snag and be difficult to move around. Short-haired puppies are able to hunt more efficiently and survive to produce more puppies themselves. This environment favors one particular trait over another, and so the advantageous trait gets passed on and becomes more abundant in the next generation. The change in the population over time is referred to as evolution, and in this example, it is occurring due to natural selection. While the implications of evolution through natural selection over billions of years are mind-boggling and awesome at the minimum, I want to focus our conversation on an incredibly relevant topic: antibiotic resistance. To discuss this topic, I want you to imagine that you contract a bacterial pathogen that makes you feel ill. So you go to the doctor, and the doctor confirms that you have a bacterial infection and prescribes you a low-dose antibiotic to be taken every day for the next 2 weeks. You begin to take your antibiotics and start to feel better over the next few days. By the end of your first week on the antibiotics, you feel completely better and stop taking the drug. No point in wasting medication, right? Unfortunately, a week in after you stop taking the medication, you start to feel worse again. You decide to take the remaining antibiotics that you had left over, but they don't seem to help. You go back to the doctor, and he prescribes you a different antibiotic and insists that you take the entire treatment regardless of how you feel.
Why do you think you begin to feel better after being on the medication for a week, but then start to show symptoms again later? Why do you think that the antibiotics were not effective when you finished your treatment in your first prescribed doses? To answer these questions, we need to apply our understanding of diversity and natural selection. Let's simplify this down and draw out some possible scenarios.
Imagine that your infection consists of just 10 bacterial cells. Each cell, while similar because they are all the same species, have some genetic differences. In all but one of these cells, the genetic variation accounts for no noticeable change. In this one cell, a mutation occurs resulting in a morphological change that makes some antibiotics less effective. Let's make this antibiotic-resistant bacteria have stripes. Now, when your doctor prescribes you antibiotics for your illness and you begin to take the medication, all the susceptible cells die off quickly, and you begin to feel better because you have fewer cells infecting you. So these cells die off whenever we create an environment with antibiotics. The antibiotic is providing a selective environment and acts as a natural selective force for any resistance to the drug. This kind of drug tolerance explains why we must prescribe antibiotics at a specific dose for a specific time frame. You must take the drug repeatedly to maintain effective doses as the body tries to filter it out, and over an extended period of time to ensure that microbes tolerant of the antibiotics are eliminated. So what would happen in the next generation? If we have a selective force of the antibiotics, the antibiotic-resistant bacteria will be the only one to survive and pass on their genetic information to the next generation. Now the population is predominantly resistant bacteria. Okay, so back to our case study. Why did the doctor have to use a different antibiotic? Well, because they are all resistant now. But if you change the environmental selective pressure, i.e. the antibiotic, then you change what is advantageous. Let's imagine that the doctor changes you over to a different antibiotic now. This creates a selective environment where this new antibiotic will eliminate any bacteria that do not have resistance. If you take the drug as prescribed, this will effectively eliminate all of the bacteria that are present that have resistance. They only have resistance to this antibiotic that was initially prescribed. Does that mean that bacteria only have resistance to one antibiotic? Not at all, unfortunately. Lots of bacteria have resistance to multiple antibiotics. In fact, many bacteria are resistant to entire groups of antibiotics, making it incredibly challenging to treat several of them.
You may be wondering how we know what an effective dose is and how long to expose the bacteria to. But you already know the answer. We use the scientific method, of course. We can match pathogens to diseases, their primary characteristics, and even test for most effective concentrations of antibiotics.
Cool fact alert! Did you know that bacteria reproduce asexually through binary fission? One cell actually splits into two. That is crazy cool! In a microbiology video later, we will discuss how bacteria exchange genetic information to create and share genetic diversity. While you would likely fully recover from your illnesses, the prognosis can be deadly if the bacteria have multiple drug resistances developed in extremely selective environments like hospitals. Unfortunately, there are only so many antibiotics available, and fewer new antibiotics are being created every year. In contrast to this, more new resistant strains are developing due to natural selection year after year. That can be some pretty scary stuff, but this problem represents why it's so vitally important that healthcare workers and patients understand how new resistant pathogens develop through natural selection, and why it's also so important that they use their prescriptions as directed.
Okay, so let's review what we learned. Genetic diversity in a population presents an opportunity for natural environmental variables to favor one individual over another. This favorable environment will lead to populations having more favorable traits as the surviving individuals pass on their genetic information. The presence of antibiotics creates a selective environment for drug tolerance and resistant bacteria. And improper usage of prescribed medications can result in bacteria that are challenging to combat. Well, that's it for this conversation. I hope you learned something along the way. If you liked the video, be sure to hit the like button below and subscribe so that you can see more of the videos that I release about the amazing world of science. See y'all next time.